introduce myself. My name is Mircea Opriș, and I am part of HBD team, and we are supporting ImmunoCAP technology in Eastern Europe. It's a pleasure to welcome you all to the second webinar in our allergy-focused series. Firstly, I would like to extend the warm welcomes to the those of you who joined us in our first session regarding the food allergies. It's great to have you back, and for the ones that are here for the first time, we're delighted to have you with us. This series, led by Professor Rudolf Valenta, is designated to provide valuable insights into the key allergy-related topics every 2 months. So today we will focus on the spring tree allergenic molecules, a crucial subject as the pollen season approaches. You can find the full schedule of our upcoming sessions in the chat. And of course, if you have been attending to our first webinar, you should also have this and receive this automatically. Once again, it's my honor to introduce Professor Valenta, world-renowned expert in molecular allergology from Medical University of Vienna. I could say that his pioneering research has significantly shaped our way of understanding allergic diseases, and we are honored to have him here today to give us the lecture. As always, we encourage you to engage by submitting your questions in the question and answer sections through the session, and by the end of the lecture, Professor Valenta will address them one by one. Please note that this webinar is being recorded and will be available for 2 weeks for those of you who would wish to review the material later. And lastly, but not least, before we begin, I'd like to mention that today we have a special guest joining us, and she will have a small but very relevant contribution to understand the topic in the best way. So please stay with us until the end of the lecture to meet our special guest. Without any further delay, please, Professor Valenta, you may start the presentation, and thank you for your time.
Yeah, thank you, Mircea. Yeah, it's a big pleasure for me to speak to all of you. I hope to make it interesting to you as much as I can, and in particular regarding the clinical relevance of the presentation. I would like to thank HVD very much for organizing this session. I find them really very useful, I would say. For me also, because I can highlight some things that I cannot just bring in a simple presentation. You may hear in the background maybe now some little bit of working noise. Yeah, it's our hospital, but don't be afraid. It's not an earthquake. We are not falling down. So everything is good, and I hope we can suppress it. As I say, it's the nice thing with the format of these webinars is that we have a lot of discussion time. So please do not hesitate to put in your questions, all you may have. I would like to highlight here a little bit some of the data. We are now very active in allergy research in the form of a bigger research cluster network. It's the Danube Allergy Research Cluster that I would like to highlight here. So if you click this link, or if you photograph it, it will bring you to our allergy research team that is concentrated actually in Vienna, but also in Lower Austria, in the surrounding vicinity at the River Danube. Papers of myself you can find in the PubMed under this link, so do not hesitate. And if you have questions, please drop also emails to me personally if you do not have time to get your questions answered as of today. Now, let me reveal to you my conflicts of interest. I think that's important. I work with pharmaceutical companies also a bit, not so much as many others do. They have a full list. But my mission is, of course, to transmit the laboratory work into clinical practice. And this is a reason why we also need the help of companies at some time point, especially also for the development of microarray technologies. Now, when we come to speak about spring pollinosis, we all know it's a wonderful time. We love it, and we start to feel it. Already when we go out in Vienna, we have a warm temperature, we have sunshine. I think it's going to be the same in most of the European countries at this moment, and also in Eastern Europe. And of course, some of you may have felt already earlier that people who are allergic start to have some mild symptoms. And this goes back to actually trees and bushes of the order Fagalis. Leading among them is the birch, Betula verrucosa, but there is also hazel and alder. These are the early flowering plants, and they are cross-reactive with birch. And there's also another little thing that I would like to point your attention to, and this is the overlap of the birch pollen season with the ash pollen season, and this is a allergen source that should be not neglected in particular regions and is often overlooked. Now, what I like to present to you is here a little bit of a survey that was done by what is called the GAL network. The GAL network was a European-funded project that tried to standardize methods. And one of the activities that they started was to pull skin prick test data from different parts of Europe and to rank them according to the frequency. And here's a little bit of a snapshot into this. And you can see here basically the hierarchy of sensitizations according to different allergen extracts that were tested by skin prick testing here in a group of asthmatic patients according to different territories of Europe. And you can easily see here that birch is a very important allergen source. It co-occurs, as I mentioned, with alder and hazel pollen. And what you may note here is that birch is particularly high in the northern regions of Europe. And as everybody knows, birch trees are highly prevalent there. And this is the reason why birch pollen allergy is so much expressed there. But we can also find it in Central Europe, even in Southern Europe. And of course, in Middle Europe, we have somewhat a kind of an average. And this is, I would say, also a little bit a diagnostic challenge because birch pollen may overlap with other pollen sources in testing due to cross-reactive allergens, a topic that we will discuss a little bit later. Now, I like to speak about birch pollen allergy, and the reason is very simple, because in the year 1989, the Vienna group was the first to clone a plant allergen, the first ever cloned plant allergen, and this was the major allergen of birch, BETV1. And at that time, we were introducing a technology that was called the allergen expression cDNA cloning, where we isolate from birch pollen basically the coding information, the mRNA, and transfer it into cDNA. And maybe those of you who do not go back so much in the history of molecular biology, it was actually close to the mid of the 80s that the enzyme reverse transcriptase was started to be used to transform mRNA into cDNA, and this was the basis for the construction of these phage libraries. Now, these phage are extremely versatile in infecting Escherichia coli, and what is a nice thing is that they propagate very highly. So when you put cDNA into this phage, you get a lot of members. And we were thinking that this is very important because we were not sure how many copies of mRNA coding for the major allergen of birch BETV1 would be in the birch pollen. And I have to admit to you that when we started this enterprise, it was a little bit in the dark because we were not sure if pollen would contain at all mRNA that is biologically active, because everybody knows that pollen is a resting tissue. So we were thinking that maybe we have to work with male inflorescences. Some ideas were also spent on using leaves and using callus from birch. So we had really a lot of ideas around. And by the good luck, actually, we then started to isolate RNA from pollen and indeed succeeded to construct such an expression cDNA library where we could use serum IgG from patients to look for clones that would code for the major allergen of birch, the BETV1 allergen. And indeed, I can tell you it was a historic Sunday when we developed the autoradiographies. We had at that time 2 positive clones. It's not 3 like it's symbolized here. That reacted with serum from a birch pollen allergic patient that we could segregate and then basically identify the DNA sequence of the birch pollen allergen BETV1 and look what the protein was looking like. Now, to cut the long story short, this is what we now know and what has been done for many of the allergen sources, like for birch, but also for cat and for grasses, is the way from the allergen source to get the mRNA, which is a highly volatile substance, I have to say. mRNA is extremely unstable. It can be easily digested by RNases. So we had to be very careful in the work, and this could only become stable when it was transferred into DNA that was then cloned into the phage. And then, of course, from the phage library, the allergen sequences were isolated, and we could deduce from the DNA sequence the protein sequence. And of course, that gave us the possibilities to prepare recombinant allergens, like is here in a folded form, to study the mechanisms of the disease, to use it for diagnosis, and of course for therapy and prevention. Now, this is a very simplistic figure here, but I do not know if you can imagine how much work it was and how many grams of pollen had to be homogenized, and then RNA had to be isolated that was intact. It took years to actually come to this stage. And that was not a very simple thing. And I also want to emphasize that at this time, we were here in Vienna, a very small research group, and we were kind of fighting with very big industry approaches, like big companies that were also interested to get the birch pollen allergen sequence first. So it was really a hot fight and a hot battle, but the Vienna group in this case won. And so it came that we published in the year 1989 the sequence of the gene that coded for the major birch pollen allergen BATW1, and this is the major spring pollen allergen. So it's relatively simple here. When we look at spring pollinosis, it's in a sense, it's BATW1, and then of course another allergen that I will introduce to you. And at that time in 1989, it was not like this that we had our mobile phones. We didn't have mobile phones at all at this time. We even didn't have computers like we have as of today with the working power capacity. We did not have the internet connections. And this was actually very difficult then to actually read the DNA sequence and then to compare it with the already known DNA sequences. You must imagine at that time, there were not so many genes cloned and submitted to databases. And the searching in the databases was not easy. So our colleagues had to go to Munich, to one of the big computers, and enter the DNA sequence that you see displayed here. And then the computer was searching for several hours. Yeah, today we have it in a minute or so from our home computer to find the homologous protein. And it was very interesting because what came out was that BATW1 in the sequence was very similar to a P disease resistance response gene. And I want to explain you this story a little bit because this P gene was actually identified when peas were infected by fungi. And in fact, then the peas were turning on the expression, the transcription of a gene that was called a PR10 gene. It was called pathogenesis-related protein because the pea was infected. And this meant that the pea activated a gene that was supposed to defend the pea. And then we were surprised to see that the pollen allergen from birch was similar to a pea disease resistance response gene. And this is, however, very important to understand when you hear today the term PR10 allergens, because PR stands for P disease resistance related. And 10 is a category of these proteins. So BATW1 was similar to a protein that is activated in the plant when the plant wants to defend him or herself. And in the birch, maybe it has to do it also, but surely it has to do it in different plant food, in vegetables and in fruits, because they also can become infected by fungi and other microorganisms. So this is a story about the BATW1 cloning. Actually, we were very ambitious. The paper finally was accepted in the EMBO journal, which is, it was at that time a very prestigious journal. Today, the impact factor is a little bit lower, but the publication has been cited over and over again. It's one of the most highly cited papers. And it is also the expression that a smaller research group could win against big pharmaceutical companies who were trying to do the same. Now, having the gene for the BATW1 allergen, there's something that you can do and you can think about, because you can use the DNA sequence. And I guess you will be familiar with the technology of the northern and the southern blot. In fact, the terms northern and southern blot come from a person whose name was Southern. Mr. Southern was a person who was isolating DNA and digesting it with enzymes, separating it by electrophoresis, and then he used DNA probes to hybridize it. In order to get hybridization, that means binding of a DNA with southern blotted DNA, you needed to have base complementarity. This means that the sequence, the DNA sequence that you offer for the binding to the digested DNA needs to be very similar to the DNA that is on the blot. Now, the point here that I want to make is that the sequence homology or sequence conservation between the genes and the proteins is at a different level. You know that the genetic code is degenerated. So that means that if you get hybridization in a DNA or RNA hybridization, the sequence homology is actually much lower at the DNA. And that means if it still hybridizes, yeah, there must be very high sequence conservation among at the protein level. So this actually, I have to say, was my first paper that I could publish, because my job that I got allocated was to use the DNA from the BATW1 allergen and to try to hybridize it with pollen RNA from older hazel and hornbeam. And you will ask yourself, what was the reason for this? Because the reason was simple to explain, because at that time, people knew that there is antigenic similarity of the allergens in birch, alder, hazel, and hornbeam. And we were asking here the question, if the gene for BATW1 has actually a similar gene in the alder, hazel, and in the hornbeam pollen. And the hybridization was supported or supposed to show this. And I have to tell you, this was really heavy work, because you have to isolate RNA from older hazel and hornbeam and then to perform the hybridization. And this is a proud experiment that I published in the year 1991. It shows you here in the lane B, the RNA from birch pollen that was very strongly hybridizing with the DNA probe from the BATW1 gene. And what you also can see is that there's hybridization with the older pollen and the hazel pollen. So what we can say from this is that there is a gene in the older pollen and in the hazel that is similar to the BATW1 allergen. And there's another thing that is quite interesting here, because here you see the hybridization with the RNA. This is more or less what is expressed in terms in the pollen at this moment. And you see here on the right-hand side, the southern blot. This is the genetic DNA, the genomic DNA that is hybridizing. And in order to hybridize it, you have to cut the genomic DNA with restriction enzymes. And all of these fragments are very big. Basically, the gene coding for BATW1 is about 700 base pairs. Here you can see is that the fragments are well above 2000 base pairs. And this means that each of these bands here must contain a BATW1 gene. And that was actually the first evidence that in the birch you have not only one gene, but you have several genes that code for the same BATW1 allergen. So it seems that the plant wants to protect the function of the BATW1 protein by having different genes available for it. So if one of the genes gets mutated, it can be replaced by another. It is also interesting because we know today that there are several isogenes for BATW1 and there's BATW1 isoforms. Some of them are highly allergenic and some of them are not. And this has been also the reason or one of the ideas of preparing hypoallergenic vaccines using hypoallergenic proteins from the BATW1 allergen that actually occur naturally in the plant. So this is a little bit of an historic excursion. And in the end, please take home the message. there is a BATW1 allergen, and there's also a gene, and they are similar in all of the trees belonging to the Phagalus order. This is alder, this is birch, this is hazel, this is hornbeam, this is also oak. And they are all related and very similar, but the mother of the sensitization, of course, is always birch because the pollen load is the highest. Now, this brings me to the next set of experiments. And this is another interesting story because at that time when we were cloning the BATW1 allergen, it was already known that birch pollen allergic patients very often have problems to eat apples or other fruits or nuts. And here Christoph Ebner tried to provide a molecular basis for this cross-reactivity. And what he did is he was using Ig antibodies from patients and was looking with these antibodies in extracts from apple and could visualize a protein, an allergen that was designated MALD1, and that's the homologous allergen to BATW1 in the apple. And this was quite surprising for two reasons. Because first of all, apples and apple trees are not related to birch trees, so they do not belong to the Phagalus. And there's another thing that is surprising is apple is a somatic tissue, but pollen is not a somatic tissue. So this means that a pollen allergen can be also expressed in the somatic tissue of a plant. And that was actually one of the first solid molecular reports of the presence of a gene and also a protein, an allergen, in basically a plant fruit that corresponds and cross-reacts with BATW1. And you can see here in yellow very nicely the conclusions. I'm always surprised how precise we were in this because it says that antigens in birch pollen and apple share allergenic epitopes. That's the basis for Ig cross-reactivity, and that's the reason why there is this cross-reactivity. And what it also says, and I'm surprised about this, is that there's also seems to be a threshold level of specific Ig antibodies, and this must be reached until you get a reaction in the patient. And I will show you data on this, what we can measure today with, for example, immunoCAPs. That is very interesting. Now, of course, when you have the genes for the major birch pollen allergen BATW1 and you have the recombinant protein, you can also study the structure of the protein. And here I have to admit that we lost against our competitors. So the group of ALK, led by Henning Löwenstein and Michael Spangford, actually were earlier than we were in revealing the three-dimensional structure of the BATW1 allergen. They did it by X-ray crystallography and nuclear magnetic resonance. And this shows the protein. You can turn it in different directions. So you can look on the protein from this side, then you will see more or less this one. And what you can see is that the BATW1 molecule inside has a kind of a hydrophobic cavity that can also take in hydrophobic ligands, as you can see here. I have today, I think nobody really knows for sure what is a real hydrophobic ligand of BATW1, but there seems to be space for one. What is important is the protein shows a mixed alpha helical. These are these curls here, and beta sheet structure. This is these sheets here that you can see. And what is very important, this structure gives the protein a fold, and this stable fold is the basis for the IgE activity of the protein. Now, we lost the fight against the pharmaceutical company regarding the deciphering of the structure, but we won the fight being the first in the world to show that you can use recombinant allergens for in vitro diagnosis of allergy. It was a second paper of myself published in the Journal of Allergy and Clinical Immunology. It was a very simple paper, I have to say. I'm almost afraid to say it's too simple because we expressed just the two allergens that we had at this time. That was the BATW1 and the BATW2, the birch prophyllin and Escherichia coli. Although I have to say this was very complicated at that time because in the 90s we didn't have so powerful expression vectors as we have today. So we used at that time the PKK vectors. And it was also very difficult to clone in the BATW1 gene. because at that time there was no polymerase chain reaction available, so we had to do very complicated construction work to get the gene inserted into these vectors. But I managed and could express here the BATW1 and here the profilin. This is the results of the patients reacting with the natural allergen extract. And this was also very nice to look at, because you can see here that the patient number 1 was reacting only with the BATW1 in the birch pollen extract. It reacts with the recombinant BATW1, but it does not react with the birch profilin, that's the smaller protein. And you can guess already here that we had a lot of fantasy to develop this combination of Western blots to test and to find out what our recombinant protein could be before we knew the sequence. So we could already tell by the profile of the patient's reactivity that, of course, this was the BATW1 and this band was corresponding to it. And this was the profilin, because the other recombinant protein was recognized by these 3 patients, and there is even a band below that later on then turned out to be a calcium binding allergen. So this was very simple, but I would say very thoughtful experiments that we were doing. Now, after this, having published this in the year 1991, something happened that was a very funny story that I would like to share with you, because I went to Geneva in the year 1993, and there I had the chance to meet people from a pharmaceutical company, and this was at that time Pharmacia Upjohn. Pharmacia Upjohn, that later became Pharmacia, Phardia, and now it's Thermo Fisher, so companies changed their names, were represented at this meeting, and I was asking one of the people there, Hans Grönlund, I said to him, Dr. Grönlund, why don't you use recombinant allergens in your allergy test systems? And it was 1993. He looked at me and said, well, actually, I also do not know, but let's test it. And this is how our collaboration started at that time. And Hans became later on a wonderful friend when he went to academics. And we were bringing our recombinant allergens to Uppsala. And the people at Pharmacia were putting them into the immunocup system like it's done here. It's, as you know, a very precise system for quantifying allergen-specific IgE, although it's rather simple. It's one sponge and it's one allergen. But when we come to the field of spring pollinosis, you will see that it's very powerful and very simple, and at the same time also very inexpensive. So we put our recombinant BATW1 and BATW2 on the immunocup system, and we collaborated. And out of this came a very interesting paper that is actually underestimated. It's a paper by Robert Movarare, and Robert was generating these BATW1, BATW2, BATW4 immunocups. And Pharmacia at that time had frozen sera from patients from 6 European populations. And we were looking how many of them would be positive with a birch pollen extract and at the same time react with the recombinant allergens. And this was a surprising finding. Well, to some extent it was not, but to some it was, because you can see that the majority of the people from the northern countries, or let's say the Middle European countries, if you want, Finland, Sweden, Austria, and France, were sensitized to BATW1. All of them were positive with the birch pollen extract. However, when we were going more to the south, Switzerland and Italy, we could see that the percentage of BATW1 positives fell down, and instead the BATW2 and BATW4 positives increased. And in the beginning, we were a bit confused because we thought, well, maybe BATW1 is not sufficient to diagnose all the birch pollen allergics that were positive with the birch pollen extract. But then it turned out, no, it was the opposite. The BATW1 is the more precise tool because people in Switzerland and Italy turned out to be largely sensitized by grass pollen and weeds. And in fact, they were just positive with the birch pollen extract because they cross-reacted by profilin and polcalcines. And in principle, they were not birch pollen sensitized. And that was really something important to learn, that BATW1 is the allergen to identify people who have a real birch pollinosis. and not the ones that are just cross-reactive by cross-sensitization. Now, many years later, as you can see, 20 years later, we were using molecular testing. We were using allergen microarrays in a big European study sponsored by the European Union, where we look in children cohorts. And there's many authors because there's many cohorts. And this paper is highly recommended for reading. And here you can find similar situation. In the northern countries, like in Sweden, Bat1 is a predominating allergen. When we go to the south, we do not find it anymore so much. For example, here in Spain, it occurs, but at a very low percentage, as you can see here, yeah. And it's also different when you go, for example, to Italy. There it doesn't seem to be very important. Here, grass pollen is dominating. When we go to Germany, Bat1 is still important. You can see it here, but there's something that pops up. And that's quite a funny thing is because I think everybody will agree there's not so many olive trees in Germany. So what is behind this story? Why is there quite a lot of olive-1 sensitized people in the German cohort? And we will discuss this in a minute. It's a very interesting thing when we speak about spring pollinosis. Now coming back to birch, as you will know, and you have understood now, is birch is not only present in the Vagalis pollen, alder, birch, hazel, oak, hornbeam. It's also present in a lot of different fruits, vegetables, hazelnuts, and in spices. And people who are sensitized to Bat1 very often have what we call the oral allergy syndrome, or the pollen-related oral allergy syndrome. So there's different terms for it. I like the term oral allergy syndrome quite a lot. And these people actually have this problem when they eat, for example, apples. So they are basically birch pollen sensitized, but they cross-react with the apple allergen. Accordingly, we call the MALDI1 a class 2 food allergen because it's not primarily sensitizing by the gut, it's sensitizing by respiratory sensitization, and there's cross-reactivity with the food allergen. It is not pleasant, it doesn't look good, but it's not really life-threatening. When you go and you cook apples, the patient will not have such a symptom because the Bat1 structure will be destroyed, and then the Bat1 allergen will become inactivated. So people who cannot tolerate fresh fruit, they can very nicely eat compote, and usually they have not a problem with it. But there's something very interesting. What can happen is that when the structure of Bat1 is destroyed by the digestion, nevertheless, there may be an activation of T cells by small peptides that come from the Bat1 allergen and are released by the digestion, by the gastrointestinal tract. And these peptides may migrate into the skin. And then birch pollen-related food may trigger, after some days of ingestion, in fact, atopic dermatitis in Bat1 sensitized people. And this article here by the group of Thomas Werfel is a beautiful article that I recommend you to read. It's from 1999, and it mentions this phenomenon that birch pollen-related food not only causes oral allergy syndrome, but it may also trigger atopic dermatitis and cutaneous manifestations. But this is not by mast cell mediated or basophil activation. Now, coming to the point, what do we learn from the measurement of Bat1 specific IgE and IgE antibodies in terms of the pollen allergy syndrome, the oral allergy syndrome? And here we conducted a very nice study where we were looking into people who complained about symptoms of birch pollen allergy. And then we have a group of people who didn't complain about symptoms, or at least they were negative in the questionnaire. Now, what can we observe here in these figures? The first thing that we can observe is that Bat1 is the dominating allergen. It doesn't make much sense to test even all the other allergens. You can already assume it. Bat1 is the key molecule to initiate the process. Algae1 is a cross-reactivity, CoA1 is a cross-reactivity, everything we see here is cross-reactivity, and it depends very much on the sequence homology. So the lower response follows more or less the sequence homology of the proteins. There is also sensitization, and we call it silent sensitization without symptoms, in some of the people who have no symptoms. And what you can see here, the Ig antibody levels in these people without symptoms are considerably lower, significantly lower than the ones, of course, in the people who have symptoms. So this means that when you have a test result, an Ig test result without symptoms, it's worthwhile continue to watch the patient because the person may increase the antibody levels and may become symptomatic. Sometimes people even consider to treat such patients by immunotherapy to prevent them from becoming symptomatic. And there's very nice studies in children having shown the effect. There's another thing that you can see here. And this is was one of the questions that we had is we were stratifying the patients into patients with allergic rhinitis, actually oral allergy syndrome. Then we had here atopic dermatitis and we had asthma. And we were thinking very much that people with birch pollen triggered asthma would have significantly higher Ig antibody levels than the people with allergic rhinitis. But we were disappointed. You can see here that's the only group that has higher Ig antibody levels than the other phenotypes of Bat1 allergy were the people with oral allergy syndrome. And this tells us what you have seen already in the Christoph Ebner paper from the year 1991, that actually the higher the Ig antibody levels to Bat1 are, the greater is the likelihood that you have also oral allergy syndrome, for example, by apple, nuts, and other PR10 containing fruits and vegetables. Another thing and another word for prediction. Many people ask for this and they say, why should we do Ig testing? Why should we bother about this? Yeah? And I have a different opinion. I think allergy testing should be done very early. It should be done already in elementary school. And this is substantiated by this paper by Marit Westman. It's a birth cohort from Sweden, where you can see that children who are 4 years old have very frequently Ig antibodies against Bat1, but they are not symptomatic. So they're just in pink, they have just the Ig, but they neither wheeze nor they sneeze. When the children become older, the children actually start to develop symptoms. And I think it's very important to detect this early Bat1 specific sensitization to basically go for early intervention. That can be, for example, by spending your vacation during the school holidays in a country where there is no birch flowering. There's a possibility of having early birch pollen immunotherapy when there is light symptoms. All of these can help to reduce the development of symptomatic Ig levels in the children and to make them progress towards severe symptoms. So I'm an advocate of early Ig testing to initiate early measures for prevention. Now let's come back now to this story of the European sensitization profile and ask the question, is the birch everything? Is the birch everything? It dominates obviously all the vagales, or is there something else? And for this, we need to drill a little bit in these cohorts and understand from where we have the samples. We have in this cohort sera from Nordic cohorts, like from Norway, Eka, from Bamse in Sweden. We have it from cohorts in more or less what you can consider Middle Europe, UK, Southern Europe. And I want to focus now on two cohorts. And this is Eka from Norway, and this is Ginny from Western Germany. And I think everybody will agree that olive trees is not the natural vegetation in Norway, neither is it the natural vegetation in Germany. It belongs to Spain. Now we test all these cohorts. You see here the number of tested sera and the number of samples. We go back and we remember that we had this unexpected result that among the top 10 sensitizing allergens in the German cohort Ginny, we found the OLE1 allergen. And you can see that it's well above 12% of the children that were sensitized against OLE1. And we have now to ask the question, how can this happen? And what is the detail for this? Now we look into Norway. And when we look into Norway, we find the expected. Bedouin is dominating because Norway is full with birch trees. 26.8% of the people are sensitized, children 15 to 16 years, against Bedouin. However, and that's now the interesting thing, 5.6% of these children are sensitized against OLE1. There is no olive tree in Norway. So what is behind this? The same for Germany, even more. 24.5% of the children sensitized against Bedouin, 13% sensitized against OLE1. So what is this? Here we come now to a very dear friend of mine, who is still very active, although in very old age, and this is Professor Gabriel Pauli. Gabriel Pauli was the head of the pulmonology department in Strasbourg University in France. And Gabriel was a dedicated clinician. She always wanted to use molecular allergology to understand her patients better, but she always came with clinical observations. And she said to me, Rudolf, I have in France a lot of ash-borne allergy. And I said, Gabriel, I do not think that this is really relevant, yeah, because ash is not really spread so much by the wind. I have my doubts. And then it was like always when Gabriel and I met, we had a debate, and almost it was a fight. She was saying so, and I was saying so. And then we said, okay, let's address the point. You bring the serum, and we test, and then we look what comes out. And then we used her serum to test ash pollen extract. And we were using recombinant allergens for inhibition. And it turned out that the Ig binding to the major allergen of ash could be inhibited by the major allergen of olive pollen in an almost complete way. And this was kind of one of the relatively early discoveries, not the first, that actually there is a almost full cross-reactivity, Ig cross-reactivity between OLE1 and the major allergen of ash. And we all will agree, ash trees are very common now in Middle Europe, also in Northern Europe. You can look around, so it's really something very common. And I have put this for you on the screen. So I think we do not have the problem of olives, except we are sitting in a certain garden, in a restaurant, Italian restaurant somewhere in the summer, where they put artificial olive trees outside. But what we have is we have a lot of ash trees. This is how they look like. This is how their leaves look like. Yeah, this is how they flower. They are, I think, to a lower extent wind pollinated, but the pollen is in the air and it can sensitize. Now, we have investigated this in a little bit more detail with, at that time, a young student, and at that time, the relatively young student was Oscar Palomares. He came from the group of Maite Villalba and was coming to us to work for a few months in our laboratory. And this is one, I think, of the very important papers of Oscar, demonstrating that OLE1 is a diagnostic marker for sensitization to Oleaceae. And this was shown by gene inhibition experiments. So here you see the inhibition of Ig binding of patients to the major allergen of olive pollen, OLE1, by ash and by olive pollen extracts. There is a complete inhibition, as you can see. There is full cross-reactivity. However, there is no inhibition by, for example, birch pollen, mugwort pollen, or Timothy grass pollen. So there is no such molecule that cross-reacts, although, as we know, there is a FLIP11 allergen that has some homology with OLE1, but it's not cross-reactive. So this corroborates and confirms the ash pollen allergen can be represented by the major allergen of olive pollen OLE1 in a diagnostic test. And my bottom message is very simple. When you want to search for ash pollen, genuinely ash pollen sensitized people, you can just use an OLE1 immunocup, and that's enough to detect these patients. I would like to extend this story now into the region of France or in the country of France, where we studied also with the chip measurements different regions. And these different regions in the HAR cohort, actually, these different regions are represented here where we measure Ig antibodies against major allergen of ragweed, birch, and cypress. You can see here, as expected, birch is prominent in the region of Paris and Grenoble. It is not in Montpellier. Why should it? It's on the Mediterranean Sea, also not in Marseille, and only very little in Lyon. And that fits very nicely to the pollen counts. Low pollen counts, low sensitization to BATW1. When we look at olive 1, we find both possibilities. We find a lot of olive 1 positivity in Grenoble, and we find it actually, despite the fact that there is no olive pollen counts in the air, but we find here a lot of ash pollen in the air, as you can see here by the pollen counting. You find also olive pollen positivity, of course, at the Mediterranean Sea, in Marseille and in Montpellier. In Montpellier, the ash is still dominating. Again, this is very nicely emphasizing olive 1 is a marker allergen for ash pollen sensitization. So please bear this in the mind. Now, summary 1 of this little presentation now. Birch pollen and ash pollen are important allergen sources in spring. It depends, of course, on your pollen calendars. You may come from different countries, but in the principle, I think for this audience, this is absolutely correct. BATW1 is the genuine marker for birch pollen sensitization. You can use it also to say that somebody is older, hazel pollen allergic. Yeah, it's fully cross-reactive. You do not need a koi one. You do not need an algae one. BATW1 is enough. 1 cup can do it for all. Important, BATW1 specific Ig levels are associated with cross-reactivity to PR10 allergen molecules from actually other trees of the order Fagalis, but also plant food. The higher they are, the higher is the chance that there is a reactivity. Importantly, there's a complete Ig cross-reactivity, almost complete, I would say, between FRA1 from ash and olive 1, and therefore you can use olive 1 as a genuine marker for olive and ash sensitization. So you can diagnose your ash pollen sensitized patients by using olive 1. It sounds strange, but this is the fact. Very simple. So if we look at spring pollinosis, it boils down to BATW1 and to olive 1. And if you want to understand cross-reactivity, you can take on board the profilins, the polcalcins in addition. Now we come to the point how to use this knowledge and what else can we do with it. Well, what sense does it make to diagnose or to differentiate between sensitization to birch and ash if we have not a highly form of specific treatment? Well, this is the case, we have it. It's allergen-specific immunotherapy, and there's actually allergen extracts available for treating birch pollen allergy and for treating ash pollen allergy and olive pollen allergy. And the point what I want to make here, in principle, olive pollen extract is also suitable for treating ash pollinosis. The important point that I want to make here is, please, colleagues, do not forget to double test your patients with BATW1 and olive 1. And the reason for this is the flowering seasons of birch and ash are very close together. And I have seen many patients in my molecular test results that surprisingly I thought are only birch pollen sensitized, but at the same time they are also ash pollen sensitized. And if you neglect this testing, it may be that you prescribe the birch immunotherapy and then you are surprised why it does not work, because BATW1 does not cross-react with olive 1 and FRA1, and therefore you need in addition the treatment for the ash pollen allergy when it is of course clinically relevant. And this is what you need of course to confirm not only by serology, but also by case history and eventually provocation. Now marker allergens are perfect for identifying the patients that should have the immunotherapy. and the extract that should be given. And we are talking now about basically two scenarios or three. The person is sensitized to birch. Then birch-borne extract can be used for treatment. Bat1 positive, birch is a good solution. The patient is sensitized to olive-1. Then olive-borne extract or ash-borne extract, if available, is a good idea. And there may be both. And then you may consider both, but you may also consider what is the more relevant, clinically more relevant allergen source. For this, you have the molecules for the prescription and for the identification. But what is also important is to look if the immunotherapy has its effect, and you should understand the cross-reactivity. Now, we have made some time ago, and it goes even back to the year 2002, the decision trees, and they are very simple. And as we have made them, I think they are valid as of today. When I come to the decision, should I do birch-borne immunotherapy? What I need is, of course, clinical relevance. That's important. Patient must have symptoms in the birch-borne season. And I need a positive confirmatory test result for Bat1. I may or may not have a positive result for a cross-reactive allergen. That's another one. But if the patient is negative for Bat1, it's usually not such a good idea to do birch-borne immunotherapy because most of the allergen extracts are mainly standardized for the Bat1 allergen. I would not include that it's possible to treat somebody who is only positive to a cross-reactive allergen with a birch-borne extract, but it would not be my choice or my first choice. The second thing is ash-borne allergy. For this, you can use the major allergen of olive-borne as a marker allergen. And the same is true if olive-1 is positive. You have a patient that is either olive-borne and or ash-borne sensitized. And then immunotherapy can be considered with ash and or olive-borne. It will work either way because of the high level of cross-reactivity. Now, I think I do not need to disclose to this audience that everything that I propose here based on the decision trees has been checked by clinicians in their regular practice. This is a kind of excerpt from a very nice evaluation paper by Joachim Sastre, who has basically confirmed in the clinical cohorts that they have in Spain this philosophy of using the marker allergen concept. It has been also confirmed in Italy, and there's several of these studies. So I think it's clear to the common sense that if you are not sensitized to the major allergen of the allergen source, you should not give immunotherapy for this allergen source. So I think you do not need clinical evidence by comparison studies. It's a matter of the clear mind to tell you that this is a correct way to do it. An important thing, and it's a matter of the cost. It's a matter of the cost because you can say, okay, I do the prick testing and I get similar results, but the problem is in the prick tests, you can have a lot of cross-reactivity and it will not give you the firm diagnosis of the genuine sensitization like the recombinant Bat1 and the recombinant olive-1 test. But of course, skin prick testing is inexpensive. But the problem is, and please have a look in this paper, here clinicians have evaluated the scenario of doing skin testing, allergen extract-based testing versus recombinant testing. And in fact, the outcome is very simple. You spend a few euros more for the recombinant testing, but you prescribe the immunotherapy more correctly. And you can even reduce the costs for immunotherapy by selecting the correct extracts. Have a look into this paper. It shows it very nicely for birch-borne, mugwort, ragweed, and grass-borne. And of course, it's also true, obviously, for ash-borne as it stands. Now, we come to immunotherapy. Today, I had my day started very nicely because I had a consultation by a patient. It's actually not a routine consultation because I do not earn money with seeing patients, but it was a person working in the hospital. And he came to me with a molecular test result for FLIPI1. And I therefore could easily recommend to this person immunotherapy with a particular grass-borne extract and a particular preparation that is rich in FLIPI1 and gives very nice blocking antibodies. Now, what I recommend to all of you when you are doing immunotherapy, I highly recommend to start checking the development of allergen-specific IgG antibodies. It is a little bit like in COVID vaccines. People were saying, oh, you are SARS-CoV-2 vaccinated, so why do you get COVID? And we actually checked for neutralizing antibodies. And we simply found that 1 out of 3 people who had received COVID vaccines had not developed or not responded to the treatment and developed specific IgG antibody responses. And this is very simple. You can do this also for birch pollen immunotherapy, and you can do it also for ash or olive pollen immunotherapy if you want, by using the recombinant allergens as we have done here. And there's something very interesting. Sometimes you need a little bit of patience until you see the development of the antibodies, because you can see here a long-term and a short-term treated group. What does it mean? This means treatment for only one course, one pre-seasonal course. So here you often see a lower development of protective antibodies, whereas in long-term treatment, this is treatment for 2 years. This is in this case Alutard. You can see much stronger development of allergen-specific antibodies. However, and this is what I would say, is when you are giving treatment for more than a half year and you do not find the development of IgG antibodies against the major allergen as compared to the baseline, this means the serum taken before the treatment, then something is wrong with your vaccine, or maybe the patient is a non-responder to this particular antigen, and that can be tested. And it can be simply tested by measuring the IgG antibodies, but you can also measure it by the inhibition of IgG binding of the patients. With the post-treatment serum, you see here ranges of inhibition of IgG binding to 60 and 70%. That's a very good result. That's something that you can see in the Immunocap ISAC. If you want to spend the money, it's useful because there you see the inhibition of the IgG binding directly. But the more simple thing is to run another Immunocap with IgG conjugate. And for this, you have basically the tests available. If we speak about spring pollinosis, what you need is you need a Petri-1 Immunocap, you need a Oli-1 Immunocap. Eventually, you may want to test Petri-2 and Petri-4 to test for cross-reactivity. You test for IgG sensitization. And once you have started immunotherapy, you start IgG development before the treatment, so at baseline. And then after one course of treatment, or let's say vaccination, it depends, subcutaneous vaccinations, one series, or half a year of, if you do sublingual treatment, you can also try to test. Maybe you will find there little IgG development. I personally prefer subcutaneous treatment because it's more potent in inducing blocking antibodies. So after one course of treatment, you should see an appreciable increase of IgG levels against baseline, against the major allergens, and this is very important. Now, at the moment, we have only allergen extracts. This is all what we have. We can debate about this very much, and I'm fighting against this with all our research work. We have developed for this a new concept of carrier-bound recombinant subunit vaccines that contain peptides from the IgG binding sites of the major allergens that are bound to a carrier protein. In our case, it's PRES. We have advanced these vaccines for grass-borne allergy up to phase 2B with good clinical success, so approximately 25% improvement over placebo, which in a large group, I think is a very respectable and good result. I think you will not reach it by pharmacotherapy with what is available at the moment at the market. So I think it's really good, but we are a small country, we are Austria, and we could not immediately progress with this. So we had to engage into new collaborations to advance our technology that showed these very nice clinical results. And this puts you on the horizon, something that I think we will manage to achieve. At least I hope to achieve it in my life, that you will have in the not too far future an immunotherapy available that consists of a pre-seasonal treatment of only a few injections, maximum 5, and yearly refreshments as compared to the current schedules. At the moment, you have to accept the upper two boxes. There's not much that we can do about it, but we are working on this and we are working hard on this. We have made recombinant vaccines not only for grass pollen, but also for the house dust mite. It's two molecules. We made it for cat. We are proud of our cat vaccines. We put three major allergens into one molecule. We call them supercat because of this. Our student likes it. The term supercat, they are really very potent in inducing protective antibodies against FeLD4, FeLD4, and FeLD7. And we have done this also for birch. And we're approaching now this slowly in clinical evaluation. And I hope that in my lifetime, I will be able to fulfill also this prophecy and give you this into your hands. At the moment, we stand with this summary, and this is very important. For spring pollinosis, please remember, BET1 and OLE1 can be used to prescribe immunotherapy to patients with genuine sensitization to birch and ash. Don't be afraid to use an olive pollen extract to treat ash pollen sensitized patients. I think it will work very nicely. BET-V1 and OLE1 can be used to measure the development of specific IgG. It's better than to do 2 years immunotherapy treatment without having success. Rather measure after half a year if something is going on. And what we are working intensively on is molecular forms of immunotherapy. We have them in clinical evaluation, especially for birch pollen. They have been developed and they are in clinical evaluation, and I hope that they will be available to all of you soon. And then you will have something more in your armamentarium. Now, with that, I would like to end here. I think the presentation was very simple. We have two molecules in the essence, so it's not expensive. It's easy to test for your patients. And before I close, I have again to express to you my cordial invitation to what I think is becoming a really, really amazing congress with Nobel laureates to come. And I hope also you to come and to attend. It's the International Congress of Immunology in Vienna. We will waltz into the future of immunology for a better health with this golden man. It's Johann Strauss. He's celebrating his 200th birthday this year. So we say in Vienna, Strauss in Saus und Braus, yeah. This is the motto, what we have. And here you find the Congress homepage, www.ios2025.org. I think from most of the countries from where you are, you can travel to Vienna in a very inexpensive way. Maybe the registration is not, but the hotels can be, but they have high quality. So come and visit us. Learn more about immunology. It's the key to future treatment and diagnosis. And thank you all for listening. It has been my pleasure here to talk to you.
Okay. So thank you very much, Professor Valenta, for your
He's sending
lecture.
me all these nice little things, these hands. I even receive a hug. I never
This
got
is a
so much.
sign of appreciation for sure
Oh,
for all
thank
your
you.
knowledge, your lectures, and sharing this with us here. Honestly, I personally found it very interesting with, I think, very high impact in every allergologist clinical practice, and I'm sure that even it was more interesting and more practical for our audience here that it's actually having the patients in their cabinets. So I think it's a very welcomed presentation, and the perspective of the future in the treatment, it's also very interesting. All of us, I think we are very interested to see more about this and hear more about this. What I would like to say before we get close to the ending, it's important to mention for all of our participants that they have the recording available for them anytime they need to rewatch or need, or even to request any specific articles that they would like to study more. We are very
Yes.
happy to share this. And now, before I will invite the participants to share with us their questions, it's my pleasure to invite and introduce our special guest. Please welcome Professor Carmen Bunu-Panaitescu, Chief of the Romanian Society of Allergology and Immunology, our longtime and well-trusted partner. So, dear Professor Panaitescu, welcome to our I know you've been here from the very beginning of the webinar, but welcome now, and we invite you to make some comments on the topics. So thank you, Professor Valenta, again. Thank you, Professor Banitescu, for being here, and let us see what's next.
First of all, thank you for inviting me. It was a great surprise even for me. And as always, it's an honor to be at site of Professor Rudolf Valenta and hearing his wonderful speech. I remembered a lot of discussion we had about another big problem of allergic patients, the one linked by ragweed pollen allergy. So we could see that in different regions in the world, we can have different problems, and how important is to know the source of allergens in order to understand what is the problem of the patients and how deep we can enter in this problem using this molecular allergology approach, which is the most modern approach connected with personalized medicine. And we always know that component result diagnosis is the way to approach modern medicine, and gave us the key of understanding why some patients are responding to a certain treatment and why the other not. So we are in an open area of understanding this. So first of all, I would like to express my gratitude to Professor Rudolf Valenta for the state-of-the-art presentation he gave, a link on this important issue of spring respiratory allergy, the one linked by tree pollens allergy, and how we can come from what is in the air on how it's happening with the patients, and how we can prescribe a better treatment for them. So this allergen immunotherapy, which is older than 100 years, it's more and more new, because the new concepts are coming as much as the science is progressing. And Professor Rudolf Valetta is known worldwide that he opened this way to a better understanding and characterizing of allergens, and also on what can we do for the patients. So we saw in the first part what are the main allergens when we speak about tree pollen allergy. Professor Valenta showed us how important it is to know about BEDV1, and how using that we can consider as a main marker for tree pollen allergy for Fagale. So we shouldn't do a lot of tests for having a clear diagnosis for patients with desensitization, and how interesting we can link the allergy of birch with a lot of PR10 allergen containing plant foods, and in oral allergic syndrome, and also that connection with atopic dermatitis. So how can this disease can be triggered in patients. And I think this is a very important topic for us allergologists when we discuss with our colleagues in dermatology. A lot of colleagues from dermatology don't consider important to know something about allergies in atopic dermatitis, but we can see that allergies are the main triggers in activating that disease. And basically that T2 inflammation, it's a characteristic of what we call allergies. Doesn't matter if it's asthma, allergic rhinitis, or atopic dermatitis. Also, it was a great presentation of the fact that the measurement of BEDV1 specific IgE identifies birch pollen allergic patient, and also that is important for the diagnosis of birch pollen associated oral allergy syndrome. So with one test, you can have a response to two important issues in a patient. And finally, that you can consider H3 allergy measured by OLE1. So one allergy explained by the presence of IgE to another molecular allergen, why they have that great strong homogeneity. So understanding the biochemical processes and the presence of different kinds of molecular allergen is helping us to understand this complex mechanism and to be more and more connected with what is the necessary and what is the main thing to do to make a good diagnosis. So having this in mind in the first part of your presentation, Professor Valenta, I would like to ask you, how do you explain the regional differences regarding percentages of patients sensitized to different birch pollen allergens? As it was presented in Finland, almost 100% of the patients are sensitized to bed V1, while in Switzerland and Italy, to bed V1 respond only 60% of them, while to bed V2, 30-40%. Is it important or not?
No, it's in principle you can say that the results with the birch pollen extract in the southern countries are false positive results. Yeah. So the bed V1 test reveals this. Yeah. So in principle, what you have in the southern countries, you have a sensitization to, for example, grass pollen, including the cross-reactive allergens. And then birch pollen contains also the propylene and the polcalcine, and then these people are positive with the birch pollen extract, and then you believe that they are birch pollen positive. Yeah, they would be also positive in a skin prick test, and then you would prescribe to a patient who is not birch pollen allergic a birch pollen extract, but the sensitization comes most likely in this case by a grass pollen extract or by grass pollenosis. Yeah. So this is why the molecular testing is recommended to decipher this situation. And it's also, of course, easily understood because the vegetation in the southern countries is different. So the birch dominates in the northern countries. Yeah. So here only the molecular testing can give you the solution. And that for us, you must think back now, it's more than 20 years. Yeah. When we see these results, we were surprised, we tried to explain it. And then when we go back to the files of these patients, we find that they are positive to wheat, they are positive to paritaria, they are positive to grass pollen. Yeah. And then they turn out to be not birch pollen allergic, yeah, so to say, or not birch sensitized.
Thank you very much. And that also make me to remember years ago when we didn't have this molecular testing in Romania, we had only the possibility to make skin prick test, and we had patients with a positive theoretically skin prick test to almost all pollens, which is not possible. And I had to send them to Vienna to make the correct testing and to have the right allergen, culprit allergen, in fact, discovered. So skin prick test maybe can have a place, but it's not.
It's in the end of the day, the skin prick test solutions, the quality is getting lower and lower, and it's more difficult to access skin prick test solutions, yeah, because nobody wants to produce them anymore, yeah. And in the end, clinicians will be lost, yeah, because they simply disappear from the market. We ourselves sometimes try to get skin prick test solutions for research purposes, and we are unable to do so, yeah. So in the end, what will remain and will be the only precise tool is the serology. And I have to say it's the test result, the sensitization is the same, yeah? And we must be very clear now, we must say that by skin prick test and by serology, we measure only sensitization. We cannot just forget about also asking about the symptoms, yeah? So the IG test result is pivotal, but we also need to have our clinical case record, yeah? So I ask the clinicians, please do not forget this. You do not treat a laboratory finding, yeah? You treat a patient. You can treat a sensitization to prevent eventually, yeah? It's like you have PSA elevated, you will study initiate some more investigations, of course, yeah, like you have in prostate cancer or so on, yeah. It's the same topic, yeah, but in principle, yeah, you have to find, we have to have a reason for the disease, yeah.
That makes me asking you another question. So how can we see this measurement of specific IgE to BEDV1 in patients which are not symptomatic yet, but how can we say that that could predict the future of clinically allergy?
I think this is especially important in early childhood. Yeah, with an adult patient, yeah, there can be different reasons. Yeah, it can be also that the patient has at the same time competing IgG antibodies as induced by immunotherapy. Yeah, that will basically protect the person. Yeah. It can also be that the patient is not exposed. Some people, they work indoors, yeah, they avoid outdoor working, yeah. So there's many different factors, yeah. So we have to also consider all the time the clinical picture, and also you must ask the patient, so can you tell me now what is bothering you most? What is your biggest problem? When is it, yeah? So you must be very careful with that, yeah. And put the laboratory test result in the context of this. Yeah, so I cannot often enough emphasize this. I have here, by the way, also a question, quite smart questions, as I think, from the audience. For example, one participant is saying, well, what happens is if I do not see raised IgG4, but I see clinical improvement. That's a smart question. What do you do in such a case? Well, the point is there's different possibilities. Not only IgG4 antibodies protect, also IgG1 antibodies protect. So maybe it's worthwhile testing IgG. The second thing is there can be different reasons why you don't see clinical symptoms. Maybe the person was not exposed. So ask the person also, yeah, where have you been during the pollen season, yeah? This is why you do clinical trials, yeah, when you have been in Spain or somewhere, yeah, okay, you for 4 weeks, yeah, or so, yeah, you will eventually have not clinical symptoms, yeah. So my answer to the first, yeah, but it's a clever question, yeah. Then one person says, what about this if only one is positive along with only 6, yeah? I say, well, as long as only one is positive, there is a reason to act, especially when we talk now about spring pollinosis, yeah, because then you will probably never see only 6 positive, yeah. Only one is enough to indicate ash pollen allergy, yeah. Then one person asks, is it possible to have low IgG after 1 year of IT, but improvement of symptoms? Well, I would say it's the delta compared to the baseline. You should see at least an increase. If you have no increase, I would say the vaccine is not working properly or the patient is not responding properly to it. So for me, this would be the reason to change the immunotherapy vaccine, if I'm honest. Only 6 would be replaced by only 7. Well, in the spring pollinosis, yeah, I would not test any of those 2, yeah. I would only rely on BEDV1 and only 1. It's, in my opinion, enough, yeah. Sometimes it's better to keep the world simple and less expensive, yeah.
Okay, so it looks like we managed to answer. Okay, we have another question now. Just
yes,
arrived.
This is a difficult question. Should we initiate immunotherapy to BEDV1 in young children and teenagers? Yeah, as a preventive measure, even though they do not have symptoms at the present moment. I would say it's delicate. Yeah, I would say we do not have the clinical evidence for it. I would think it will work, but I would rather await the clinical study that addresses this. We have the clinical studies that show that you can prevent the progression from rhinitis to asthma. We do not yet have the studies for this, so I would at the moment say no. When at the latest we can start birch-borne immunotherapy? The new qualified patient from Central Europe late January. It depends on what vaccine you are going to use, yeah. This is really the key thing, yeah. We have tested several different vaccines and schedules, yeah. Some of them have a lot of build-up injections. Yeah, I think it's too late to start in January. I would start at least in November, December. Yeah, because you will need several administrations, even with the low dose. There's one very short course vaccination protocol. I do not want to say what the preparation is. I think it's 3 or 4 injections, and it's available for other pollens, but it doesn't work at all. You can read the literature and then you know what I mean. Yeah. The other ones, they require more build-ups. Yeah. And so January is, in my opinion, too short. Which patient group should be strongly considered for testing for indoor allergens, even if they experience symptoms only in spring? To be honest, if a patient has only symptoms in spring, I do not see a reason to test for indoor allergens, yeah, if I'm honest, yeah. So why would you do this, yeah? Should we exclude nuts, vegetables in BET1 sensitization and IgE cross-reactivity? To be honest, if I approach the question, yeah, of fruit and food allergy, yeah, it's a bit more complicated. BET1 is a good indicator of oral allergy syndrome, yeah. You can have also oral allergy syndrome due to profilin. You cannot have it because of BET4, because BET4 is only a pollen-specific protein, yeah. When you want to target testing for fruit and food allergy, it may be a good idea to include lipid transfer proteins in addition to profilin and BET1. Yeah. So if you want to be really solid, yeah. But when you have somebody with an oral allergy syndrome without very severe symptoms, in my opinion, the BET1 is enough. Lipid transfer proteins usually cause more severe symptoms. Yeah. What other preventive measures should we advise patients to take when having BET1 positive and no symptoms besides eviction, as you said, but they do not develop other pathologies? If we should not start, well, I think if you have these school holidays, it can be a good idea to cut the birch pollen season as short as you can to place in a holiday, yeah, that brings you to a completely different part where there is no birch pollen exposure. There's one thing is a birch pollen exposure very strongly increases the BET1 specific IgE levels, and they load then on the effector cells, and this makes the kids more and more sensitive by the time, yeah. So I think the more time you can eliminate from exposure, the better it is. And I think it's really hard for birch pollen because, I mean, for birch pollen, the season is relatively short. You can cut away easily a few weeks, and I think that can be quite effective. This is what I would try to do with my modest knowledge, yeah, so to say. Now we have no
Great.
questions.
We have appreciation for sure. Thank you very much for responding to all the questions. And we're happy to come to an end, I think. And again, I would like to thank Professor Rudolf Valenta for his lecture and Professor Carmen Bonobanitescu for her contribution here. Very valuable and interesting things have been added to the discussion. Thank you for all of our participants for the time and for your interest here. We are very happy to have such a great audience. And if there is no further questions, we can end this session. And of course, all of you are invited for the next ones that are willing to come.
Yep, and the next time I should say, I think maybe we will speak about grass pollen. There I have some very concrete things even here that you can refine your molecular tests with certain vaccines. Yeah, for this I have some special little recommendations.
So there is absolutely no reason to miss the next one. No reason. Okay. Thank you, both of you, again for the time and the sharing your knowledge with us. And it's time to close this webinar and wish you a nice evening to all of us.
Thank you very much for hosting us. Yeah. Bye bye.
are welcome. Thank you very much. Bye bye.